In the name of Allah, the most gracious, the most merciful. Assalamu alaikum. Today we will discuss about leaf anatomy in Selaginella, life cycle of Selaginella, which includes asexual reproduction and gametophyte or sexual reproduction. So let's start. This diagram is showing the leaf anatomy in Selaginella. The upper and lower surfaces of leaf are covered with upper and lower epidermis. These are one cell in thickness and their cells contain chloroplast. The stomata are present in both upper and lower epidermis. In between epidermis, mesophyll tissues composed of thin-walled parenchyma cells are present. The cells contain chloroplast and enclose air spaces. The vascular tissues occupy the center of the leaf. Xylem is composed of tracheids only, and phloem comprises of a few sieve cells and many parenchyma cells. Phloem surrounds the xylem. Life cycle in Selaginella. The life cycle in Selaginella is characterized by alternation of generations. Both the spore and gamete producing generations are independent. Sporophyte. The sporophyte reproduces both by vegetative multiplication and spore formation. Vegetative multiplication is caused by three methods: adventitious branches, tubers, and resting buds. Some Selaginella species reproduce vegetatively by development of adventitious branches, which separate from the parent plant and develop into new sporophytes. Other species produce tubers during unfavorable conditions for growth. These tubers develop into new plants on return of favorable conditions. Similarly, some species develop resting buds at the ends of aerial branches. These are surrounded by leaves. These can survive adverse conditions and develop into new plants when conditions are suitable for growth. Asexual reproduction in Selaginella. All Selaginella species are heterosporous. It means they produce morphologically and functionally distinct spores of two different kinds, the microspores and megaspores. The microspores are smaller in size and produce in microsporangia, while the megaspores are larger in size and produce in megasporangia. What is Sporangioferous spike? The sporangia are produced in the axils of ligulate leaves called sporophylls. The sporophylls aggregate and form definite structures called strobili. The arrangement of sporophylls in a strobilus just like bracts in a spike. Therefore, the strobilus is also called Sporangioferous spike. The structure of sporangia in Selaginella. The microsporangia are small, stalk, variously shaped, mostly oval. Whereas the megasporangia are large, stalk, four-lobed, and normally present at the base of the strobilus and contain a few spores of larger size. Both microsporangia and megasporangia have two-layered sporangial wall surrounding tapetum and sporogenous tissues. The tapetum develops from innermost layer of the sporangial wall. Both sporangia differ in size and position in the strobilus and number and size of spores. Both sporangia form a vertical cleft in the wall and release the spores. This is the microsporangia, structure of microsporangia, and this is the structure of diagram of megasporangia. And microsporangia contains a lot number of microspores, sporangial wall, and ligule. But the megasporangia contain a few spores of larger size, also have the sporangial wall and a ligule. The gametophyte in Selaginella is produced by sexual reproduction. The microspores develop into male gametophyte, while the megaspores develop into the female gametophyte. The germination of spores is precocious. It means it starts within the walls of their respective sporangia. The male gametophyte is released at 13 cell stage, whereas the female gametophyte comes out at various stages of development depending upon the species. Male gametophyte. A microspore is minute, tetrahedral, and uninucleate. Its protoplasm is surrounded by two-layered wall. The outer wall is called exine, which is thick, and the inner wall is called intine, which is thin. The microspore divides by an unequal division to form a small lens-shaped prothelial cell which does not divide further, and a large antheridial cell which divides by severe divisions in various planes. This results in the formation of a sterile 8-celled jacket, enclosing 4 primary spermatogenous cells. Each spermatogenous cell undergoes 6 successive divisions to produce 128 to 256 sperm mother cells. Each sperm mother cell metamorphoses into a biflagellate male sperm. The jacket cell disintegrate and the spore wall break to release the male sperms. This diagram is showing the different developmental stages of male gametophyte. In a part of this diagram, there is a microspore which have two walls, the outer thick wall exine, the inner thin wall intine. Then in the next stage, the microspore is divided into two cells, a small prothelial cell and large antheridial cell. The antheridial cell undergoes further divisions and produce 8 jacket cells and 4 primary spermatogenous cells, which undergoes 6 successive divisions to produce 128 to 256 sperm mother cells. Each sperm mother cell undergo metamorphosis and produce biflagellated male sperms.